Hi listeners, this is Maxime speaking, and welcome back to our podcast of theoretical biophysics. I have the pleasure of welcoming Alma Dal Co, who's coming from University of Lausanne. She's currently starting her group and made a beautiful study of microbes in a small chamber and measuring stuff. I'll let you the suspense to discover what she did. And I guess you can hear around us the birds are singing. We are on the terrace of the ME conferences in Plön, and that's why we are together to talk about these microbes today with Alma Dal Co. Hi Alma.
Hi, thank you very much for the introduction. So I'm glad to be here, and I would like to tell you today about something that is really close to my interest, which is trying to understand how complex biological systems work. And particularly, I'm thinking at microbial communities. So microbial communities are often composed of many species, and they produce processes that are very relevant to humans. For example, they cycle the elements. They fix nitrogens that is used by plants, or they even produce vitamins and other compounds that affect our health, just like drugs that you take from your mouth. So those communities are very important. That's why there's increasing interest in the last decades to study them. And I see those communities as systems composed of parts, the single cells, that interact in space through a network of interactions. And this is what I do during my science. I try to understand how spatially structured microbial communities interact. And this is generally a difficult question in nature because there are so many species and you don't even know the many ways they can interact. They can exchange metabolic compounds, but they can also, for example, harm each other via cell-cell contact. So there's many types of interactions, and you have to start somewhere. And what we do is that we simplify the systems. So we work with what's just called synthetic communities. Synthetic communities are a representation of simple nature, if you want. So we take either isolates from the environment and assemble a community, or we even engineer microbial species to engage in certain interactions. And this is what I typically do. I engineer interactions in communities, and I study simple communities composed of a few species. So today I will tell you about this work. In this work, what I did was that I studied interactions between two bacterial species. And I asked the question, can we grow these two bacterial species in space and understand what is the spatial interaction network that occurs within these spatially structured communities?
Yeah, this leads to beautiful images of bacteria growing next to each other. So those bacteria, they are rod-shaped, and you can label them with different colors depending on who is who. And it gives some beautiful images like a carpet of rod shape which are growing with time. And so what did you extract from that?
Yeah, so the information we typically get is resolution of individual cells. So we can still measure the single cells and their traits, like how fast they grow, and even their gene expression if you want. So we have a very nice resolution of the single cells when we look under the microscope, but we also can see many individuals of these spatially structured communities, typically 1,000 cells. And to have this resolution of individuals, but also communities, let's say these 1,000 cells growing together, what we do is that we developed a technique that is based on microfluidics, that allows us to grow these 1,000 cells in confined environment where they cannot escape, and they create what I would say is a two-dimensional community. So they grow in chambers that are as high as a cell and as big as 60 by 60 micrometers. So 60 by 60 micrometers is it's small. It's like a little bit bigger than a cell you have on your own skin, you know? So we're talking about a very small environment, but bacteria are even smaller than your cells. So there's 1,000 of them in this 60 by 60 micrometer chamber.
So basically you can align next to each other like a 60 cell in this square chamber, which is open to one end, if I see it correctly.
Yes, it's open on one end. So overall, the population that fits in the whole chamber is 1,000 cells, and as they grow, the two cell types that we grow together, as they grow, they are slowly moving out of the chamber because the chamber has a fixed size, obviously, and so we have constant density, constant population sizes, but as they grow, they push each other out of the chamber and they're flushed away. So we have this nice control system with fixed population size. But obviously the composition of these two cell types might change in time, and this is also what we see. So what are these two cell types doing? We have engineered these two cell types, which are Ecol, by the way, so a very common bacteria that you have in your gut. And these two cell types cannot produce one fundamental amino acid each.
Some food.
Yes. So and it's not even food, it's actually building blocks. So amino acids are components of proteins. So they need to build these two amino acids, and along with many others, to build their own biomass, their own body. So what they do is that they effectively cannot produce a fundamental amino acid. So alone they cannot grow because they cannot produce more of their body, the material they need, these proteins. But when you mix them, they can. And why can they do that? Because they leak out, and this is very common in the bacterial world, they leak out some of these amino acids in the environment. This is passive leakage. It goes through the cellular membrane because it's just more costly to evolve ways to avoid that. So there's a little bit of leakage of these amino acids into the environment. And whatever cell is close by is going to take up this precious material. So these two cell types are grown together, and each is leaking out all amino acids, in particular the one that the partner type needs. So when you grow them in co-culture, they are able to grow and exchange the amino acids. And if you're curious, these two amino acids are proline and tryptophan.
Yeah, and so we've got this beautiful image of a carpet of cells aligned to each other in the chamber. One is red, the other is green, and the red one is not able to produce the tryptophan, and the green one is not able to produce the proline. And so we've got this kind of sectors in the community, which seems to be dynamical. I mean, if you play a movie with that, the cells are slowly moving up to the exit of the chamber because they push in this direction. And so, very intriguing, this formation of sectors. Can you tell us a bit more what is ruling the scaling and the formation of the sectors?
Yes. So as we, let time pass, these two cell types exchange these amino acids, and as you correctly see, there are patches that are formed of the two cell types. And what we see is actually that one of the two cell types becomes a bit more rare, and this does not depend on the initial condition of your community. It really goes to an equilibrium. Each of these communities, we have replicates within different chambers. Each of these chambers will go to an equilibrium fraction of the community. That is not 50-50. It's 20-80. And this is actually a balance between the leakage of the amino acids and how much it is needed of that particular amino acid. So they are exchanging these amino acids, but the equilibrium community is, asymmetric because of this balance between how much is leaked and how much is needed. And indeed, what we see in those images with time is that you see many less red cells. They stabilize to 20%. So there are smaller patches of these red cells, also because they're more rare in the community. Now that we have these beautiful images where we see these spatial patterns, these clusters of red, these clusters of green, all intertwined, we could see that actually the location of the individual cells determines how fast they grow. So we could do that because we analyzed many movies, and we saw that typically cells would grow faster when they are close to the partner type. So if you take a red cell surrounded by many red cells, this focal red cell cannot retrieve the amino acid it cannot produce because all the neighboring red cells have the same problem as her. They cannot produce that amino acid, proline in this case. So after tryptophan actually.
Tryptophan.
But after a while, what you see is that if that cell moves close to the partner type by some reason, or if you analyze cells that are already close to the partner type, those cells are growing faster. So the distance between the two cell types determines how fast cells will grow. But this is just a preliminary observation, right? That you need to be close to your partner to be able to receive the amino acid you need and therefore grow fast.
This gives a hint for a deeper understanding.
Yeah, indeed, what we did then was to go further and really quantify this. And what we were after exactly was to try to understand what is the neighborhood that best predicts a cell's growth rate. So we can measure growth rates and we can measure who's around a specific cell. And we want to know how far away from a focal cell. We have to analyze the community in terms of presence or absence of the partner to be able to predict the focal cell's growth rate. For example, it could be that for the cell's growth rate, it only matters the immediate neighbors. Only those are the ones that may give this cell the amino acids. Or it could be that it's a much larger neighborhood. So we went and measured this, and how we did it was to plot the growth rate of many cells against the composition of the community, or better, the presence of the partner type within a certain fixed distance. And what we find is that there's a correlation that is positive. So cells tend to grow faster when they have the partner type present in this fixed distance. And we find there's a positive correlation that we can quantify. But now the interesting thing is that if you vary this distance, you can have different strengths of correlations. And you will hit, by repeating this analysis iteratively, you will hit a certain distance that if you analyze the composition or the presence of the partner type within that specific distance, you have the highest correlation between the growth rate of the focal cell and the fraction of the partner type within that distance.
So it gives you the scale of the interaction in a sense?
Yeah. This is, well, the definition is that this is the neighborhood that you have to analyze to predict a cell's growth rate. But in other words, is really the neighborhood that the cell mostly perceives, or from which it retrieves the amino acids that set that cell's growth rate. So we call this distance interaction range, because it's the interaction, it's the range at which cells exchange these two amino acids. And this was, I think, the first work that really quantified interaction ranges at the level of individual cells. And we found something absolutely surprising. So look with me at those images. And I already told you that these chambers are 60 micrometers times 60 micrometers, so there's 1,000 cells. But when you look close by, the cells do not care about the whole community.
Indeed.
They care only of a very local and small world around them.
So what we see here is an image of a community with this red and green bacteria, and is drawn around a focal strain, a bacteria of interest, a circle which actually corresponds to the interaction range, in a sense, the neighborhood that is sensitive for this particular bacteria we are interested in into the community. And what is striking here is that the red cell and the green cell doesn't have the same range of interaction. It seems the circle around the cell that they sense is not the same.
Yeah, indeed. We found out that for one of the two types, the interaction range was 1 body length, so 3 micrometers. This is very short. It means that only the first neighbors effectively determine that cell's growth rate or are exchanging amino acid with that cell. For the other cell type, this was slightly bigger. It was 3 times the body length, 3 times that size. But both interaction ranges, which I said correspond to 3 micrometers and 12 micrometers, both interaction range are really small.
Indeed. And actually the beauty of this work, to my opinion, is really you get to quantitative data like this. It's not a qualitative thinking, you really have a number where you can rely in further analysis.
Yes, so this was for us and for me particularly striking, because being a physicist, I started thinking of order of magnitudes and, back of the envelope calculations, and I thought, oh my God, but an amino acid inside my little chamber of 60 by 60 travels from one side to the other based on the diffusion constant that we know in 2 seconds. So why is this little chamber with my 1,000 cells not an homogeneous environment?
Definitely.
Why do cells care only of their immediate neighbors? So it must be that the landscape of amino acid concentration is very rough. It's not homogeneous at all.
So what shaped it?
That was our next question. And to be able to answer this question, which is what shapes the interaction range and what makes this landscape of amino acids so rough. For answering this question, we needed to go to mathematical modeling. And what we did was to represent with an individual-based model what was happening in our community. So we have 2 cell types, and the growth rate of each of the 2 cell types can be calculated from the amino acid that is limiting that cell type growth rate.
Okay, so basically the more you got this amino acid, the higher is your growth rate.
Yeah, exactly.
Okay.
And so now we only care of the diffusion of these 2 amino acids in space, their production, and their uptake, right? So we did a model where we were first having a grid of the 2 cell types. So there was a grid that represents the individuals. And below that, there's a grid, so a 2-dimensional grid, where we represent the concentration of the 2 amino acids. And the concentration at any location in space can vary because cells are taking up the amino acids or leaking them down in the environment. And in the environment, they diffuse.
So you got in the equation, if we start to think about equations, a source term, a sink, and a diffusion process.
Yeah, exactly.
Okay.
Exactly. So we include all these terms of uptake, of leakage, and of diffusion, and we can write down equations that describe how the amino acids are traveling around in the community, being taken up, being secreted, and then diffusing between, grid points and between locations. So now, if I give you a certain arrangement of the individuals, you can really take these equations and calculate the landscape of amino acid, or the concentration at every location in point of the 2 amino acids. And so now that we have this concentration, it's quite easy to calculate the growth rate of the individuals, because as you said, the growth rate is really a function of the 1 amino acid that is limiting your growth. So now for every individual on our grid, we know the growth rate. So we have some prediction of the growth rate based on really biophysical processes of diffusion, uptake rates, leakage rates. So we plug in those rates that are found in literature, and this is the advantage of playing with Ecol that is so well known. So you know the uptake rate of proline, you know also the diffusion of proline, that's independent from the cell type, of course.
So in a sense, there is no fitting parameter in this model.
No, we did an estimate of the leakage based on equilibrium fractions.
Okay.
Yeah, but the leakage is actually not setting the interaction range. This is what we found. So,
interesting.
So this was a beautiful thing that we discovered.
Yeah.
But okay, this goes into a longer discussion. We will need to involve the audience with paper and pen. That's unfortunately. But okay, so now we are at the stage where we have prediction of growth rate from those parameters of uptake, diffusion, and leakage. And we use this predicted growth rate to do the same analysis that we did with the measured growth rate. So this, correlation between growth and fraction of the partner type within an interaction range. This is the correlation analysis we did with the measured growth rate.
So with your model, you mimicked your experiment.
Yeah.
And so did it match?
Yeah. So what we found is that when you do the same analysis as with the real growth rate, well, you find the same correlation patterns that show that there's an interaction range, and that this interaction range is short for the 2 cell types, and it's equivalent to about 2.5 and 11 micrometers. So we recapitulate very closely the 3 and the 12 micrometers that we found in the experiment.
Definitely, that's accurate actually.
So what we found is that we can predict from molecular exchanges parameters that are biophysical, biochemical, so stuff at the level of the molecule, we can predict the behavior of individuals. How far do they interact, how well they grow in communities. And this is really fascinating because I think one of the hardest things in biology is to understand how different scales of organizations are connected. And what should you retain of a lower scale of organization to the next scale of organization to be able to understand the scale.
Totally.
In physics we often do that. Yeah, you want to know, what temperature is. Ultimately is velocity, but sometimes for a certain problem you don't need to take care of the velocity of the particles, you just need the temperature, and that's going to predict something about a larger scale system. And here we're doing the same. And what we found actually is that of those molecular parameters that were the ingredients of our model, there's actually one that is particularly important and is setting the range at which individuals interact.
Which is?
It's the uptake rates of the two amino acids.
So the rate that they eat or absorb the proline or the tryptophan. Okay.
Yeah, indeed. So what we found out is that what sets the range of interaction is the ratio between the uptake and diffusion of the amino acids. But now, the diffusion of amino acid is actually pretty constant across, the various amino acids, and is also similar to the one of glucose. So those molecules don't have dramatic differences in their diffusion constant. So this parameter cannot justify the difference in interaction range we see, because it just doesn't vary as much. On the other hand, the uptake rate of amino acid, that really varies. And there's good reason for which it varies, because certain amino acids, like tryptophan in our case, are so precious that there's a lot of incentive to take it up at a very high rate from the environment. Whenever it's present, you want to take it in, because producing that amino acid yourself is very costly. And indeed, the uptake rate of tryptophan is much higher than the one of proline. And that's why the interaction range of the cell type that requires tryptophan is much shorter. You see the point? Like, if you throw in the environment a molecule, and this molecule is taken up at a very high rate, well, that molecule will not travel very far from where it is produced. It's going to be immediately removed and taken away. So the interaction that that molecule mediates is actually a short-range interaction. So high uptake rate, short-range interactions.
And you got it quantitatively, which is a real performance, to my opinion.
Yeah. And, for METI-heavy users, we also go further and actually predict from those same rates what is the expected equilibrium fraction of the community, what is the expected patch size of the two cell types. And you can really go then the next level up. So we went from molecules to individuals, from uptake rates of molecules to interaction ranges between individuals. But you can do a step further and calculate what is the global community fraction of the two cell types. What is the average patch size of one type and the other type? And even what is the community growth rate as a whole? And you can do that with more maths. And using what is known as pair approximation, which is something that physicists use a lot to study spatial systems with equations that are sometimes analytically solvable. And we actually can solve those equations. But this is maybe a next chapter.
For sure. But then, if I may do two comments on this work, which is amazing. I mean, bridging the two scales, single cell and scale of the population, is really, really informative. And I'm thinking now that you are explaining to me this work, measuring patch size and the population fraction is quite easy actually. You take a microscope, easy quote. It's not that easy, you need to work to have this kind of images. But in the end, since you've made the mass and basically given the mechanic that bridged this molecular scale where uptake is important to the pattern of microscopic observation, you could definitely quantify many uptake rates of unknown amino acids or different conditions just by analyzing your images you can take in this microchamber. I think it's a really powerful approach that, to my opinion, might be very interesting for microbiologists in the field.
Yeah, I think there's very many advantages of having found some equations that reproduce the data. Also, just we study a particular system, right? But when you see that very general mechanisms like the uptake and diffusion of molecules can describe properties that you measure in your system, this means that you're after generalization. That there are some general shared parameters that you know are important to determine individual properties like interaction ranges and so on. And these interaction ranges are very important, and these individual properties are very important because ultimately is the sum of all the activities of the individuals, all those interactions that determine what our overall community is doing. For example, in our case, we could see that short-range interaction will lower the growth rate of a community because these two cell types need to exchange amino acids. And if you have short-range interaction, very often within your interaction range, there are cells that are similar to you, which do not offer you the amino acids you need. So if they would interact at a larger range, everyone would share better those amino acids, and the growth rate of the population would be, of the community would be much higher. And this can go beyond just growth. It could be that two cell types need to do some particular chemical process, and that enzymes are shared or chemical compounds are shared. And again, the range at which these chemicals are shared is gonna determine the productivity of the community with respect to that specific, chemical you want to produce, for example. So it's very important to understand how individual properties affect the overall productivity, the overall processes that a community can perform.
Yeah, that's amazing to have this mechanic available now. And maybe the second comment I wanted to make about this work, where I see really the importance of the physics in the field, is that the uptake parameter is a ruling parameter of the dynamics, and you show it beautifully. But what is changing the uptake parameter doesn't really matter in the end to define what's happening at the larger scale. So maybe the uptake parameter can be modified due to mutation in, I don't know, a porine which allows to import the proline, or a mutation in other functionality of the cell. And actually having this simple parameter that can recapitulate a lot of modification that the cell can undergo is very powerful to understand actually what matters in the system.
Yeah, I share this perspective. I believe overall that if you are able to see that a molecular parameter, something so specific like uptake rate, has so many consequences for the ecology of the community and processes of the community, you actually can start connecting the ecology and the dynamics of the community to some evolutionary questions. Because evolution does not act on the community as a whole, acts on that small parameter. So if you see that evolution can target this parameter, you then connect the action of evolution on this parameter to the changes of the community. And you can better understand this fascinating problem of how evolution affects ecology and vice versa.
Yeah.
And maybe it affects through a simple parameter.
Who knows? And so we've got a couple of minutes left. Is there any perspective you want to share with us on this work, or?
So I'd say that this specific work told me that cells were growing better when they were surrounded by a different cell type. And maybe this is not always true, but I think this is very true for humans. I think that really being surrounded by people that are exactly like you is not a good idea. So that's why I believe not only in research it's good to interact with people from different fields and perspectives, but also in life to open up to these differences. And so I think it's overall not good to interact with people that are just like you.
I totally agree on this conclusion. So thank you very much, Alma Dal Co, for having this little chat with us today. And I guess if you want to follow up with Alma, I should be very pleased to receive your mail at the University of Lausanne. As I said, she's just started her group, so she's very ramping up to very exciting researches. So thank you very much, Alma.
Thank you very much. It was a pleasure to be here.